Good day everyone, welcome to the course on biological data analysis and visualization with R. In this week we have discussed ChIP-seq data analysis. So, as we said, we will discuss these analysis steps with two different data sets. In the last few classes we have worked with BAM files. So, we have started the processing with BAM and then we followed the full pipeline until the peak calling part as well as the visualization. So, we finally ended with visualization where you could see the count data or the read distribution across a specific genome location, as well as we saw the results of the peak calling. So, this is something that will follow when we have our own data set, right. We generate our own FASTQ files, we go through the full pipeline and we get the final output. Sometimes what we see is we also have another type of files that are available to us if we are looking at other data sets. These are bigWig or BW files, okay. And this data set 2 is actually the analysis will start from these BW files and we will see how we can visualize the results, etc. It is also possible to do peak calling from BW files, but they are not very easy to do because a lot of parameter optimization is required. So, this is something what we will be covering in this class. So, we will talk about ChIP-seq data analysis or visualization when we have this bigWig and BW files, right. So, what would be the steps? Okay, so, first I will discuss what is these bigWig or BW files, and then we will talk about this how do we visualize them in using different tools again in R. Okay, so, we have seen this flowchart already. So, we have talked about the left part. And then what we are going to talk about now are these the right part, right, where we start with a BW file and we actually visualize the results. So, these BW files, they can be converted from bed graph file and they can also be converted to bed graph files. Okay. So, we can convert bed graph files that we generated in our earlier example, earlier data set from this binning experiment, right, binning analysis. We can convert those data to bigWig or BW files, okay. And we can also convert these bigWig files to bed graph files and we can visualize. So, the advantage is, of course, the bigWig files these are binary files, whereas the bed graph files we can actually visualize, we can do some filtering as we have seen already. Okay, so, the data set that we will be working with, this second data set is this, here is a link, and as I briefly mentioned, this is an HTK 27me3 mapping in mouse epithelial cells. The control is the input DNA. Okay, so, very briefly the bigWig files that will handle, right. So, bigWig is a format for display of genomic data in a genome browser track like UCSC genome browser track. These are dense continuous data and in binary format, okay. And the advantage is, of course, these are smaller files than the BAM files, right. So, as we have seen, when we start with BAM files, these BAM files are really big because they contain all this information about alignment, whereas these bigWig files or BW files, these are much more dense, okay. Only the regions where we have some read information, these are stored in these files, okay. Okay, so, how would we proceed? So, we will start with these BW files and then we first convert them to bed graph format using this tool called bigWig to bed graph, right. Again, this is some of these tools that are already built in here. So, it is a Linux tool, right. In this link you can find this tool, you can download and use this tool to convert bigWig files to bed graph files. And then we also need the genome annotation, right. So, as I said, for this experiment this is based on mouse cells. So, and the annotation that was used is the MM9, right. So, this genome build was used, that was MM9. So, we will use that bioconductor package BS genome musculus UCSC MM9 version, and we also need the biomart, right. So, to connect to ensemble, and we will also use these packages genomic ranges, I ranges that we already used in the last class. Then we will visualize the results, right. The data tracks, again very similar exercise as before, where we will take this bioconductor package rtrack layer and GFIS, okay. As I said, we can also do peak calling, right, after we have converted BW files to bed graph format, okay. This is possible, right, to take this bed graph format and we can actually do the peak call. right, very similar to what we had done after the binning experiment there. Okay. So, pick call, this is done with this max3 BDG pick call. Okay. I will show you this function in a moment when you go into the hands on, but the issue is you need a substantial parameter optimization, right. So, if you just run with default parameters, you might not get the right results. Okay. And this is the reason why we always prefer to do pick calls from our raw data, right. The raw data BAM files, we take the bed, we convert them to bed files, and then we take that data file to do the pick calling. Okay. So, this is something that we have done in the earlier exercise. If we go back, we see the R code. Okay. So, what we are going to do is we first see, right, how we can convert bed graph to bigWig format or BW format. Let us say you have to submit your data for a report, etc. You can always submit the data in the BW format, so that we can visualize this, and these are also smaller in size. And then we go into the BW file processing, right. How do you convert that to bed graph? How do you visualize the results? Okay. And I will also show you the function that you can use to actually do the pick calling from BW files. Okay. So, let us now move on to the hands on part, right, and this is the last hands on that we did, where we have these results, right. So, we have this beans 500 dot bed graph. We can generate BW files or bigWig files from these bed graph files. Okay. So, that is what I am going to first okay, let us see. Okay. So, start.bed graph, right, it will list the bed graph files that we have. The other thing we can also do is we can say this minus L, so that we can see the size of these bed graph files. So, here are the size of these files. Okay. So, we can convert these bed graph files to bigWig, and here is the command, and again we have another tool, right, which I will show you in a moment, where we have these tools in this link again built here, right, from UCSC for these conversions of different formats to each other. So, here we have this bed graph to bigWig, right. Simply clicking this, we can download, and similarly we have bigWig, right, bigWig to bed graph. Okay. So, interconversion between these two formats is possible. Okay. Then we also have other tools which we will not go into, they also have specific functions. Okay. So, we will use these two tools, one is the bed graph to bigWig, and the other is bigWig to bed graph. Okay. So, let us see if we have this tool downloaded already. Yeah, we have bed graph to bigWig here, as you can see. So, we can simply run this here. Okay. Okay. So, this is executable. So, I am running this as dot slash, and we can convert these bed graph files, right. After binning, we exported the data in bed graph format, and okay. So, it is saying DM6 chromosome sizes we need. Let us see where we have them. All right. So, we need the DM6 chromosome sizes. So, these chromosome sizes are available again here, somewhere here. Let me see. Okay. Let us see if I have downloaded it, otherwise we have to download again. Okay. Okay, no. Okay, let us see if we have somewhere here. Okay, so I don't have them here. So let me search first where we have this, because we need these chromosome sizes here. and yeah, so we have this here. Okay, it's not this one. So again we have this in here, I think. Yeah, so in again we have this in this like a related path, we have these chromosome sizes, right. So we need this chromosome size file to actually convert this bed graph to bigWig. Okay, so we'll just simply download. And we'll put in this path here. Okay. Okay, so we have the chromosome sizes now, so we can run this. So we need to mention this chromosome size file. So we need for whichever organism we are working with. Okay. Okay. So we need to call this as an executable. Okay. So this is the final command, right? /bed graph to bigWig, control bed graph file, the bed graph file that we want to convert into bigWig format, the chromosome sizes for the organism, so DM6 for Drosophila, and then we have the output file name. Okay, so this is the format, and similarly we can convert each of those, right, files that we exported after binning into the bigWig format. Okay, so we can now see whether they are in this format. Yeah, so you see that we have now converted this bigWig format. Okay, we can also see their sizes, and you'll see their sizes are smaller. Okay, so if you want to compare, right, we can also see the sizes. As you can see, the sizes of these bigWig files are much smaller, right. So this is the advantage of storing data in bigWig format. Okay, so we now understand, right, how we can actually convert this bed graph to BW format or bigWig format. What we are going to do now is work with another data set where we have downloaded this bigWig format, and we'll see the data. Okay, and as I mentioned, this is the data, right, and we have two data points, right, here, and one is input, another is H3K27Me3, right. So this ChIP experiment has been done, and the first one is input chromatin. Okay, and these are this file we can download from this link here, these BW files. Okay, so that's what I have done, and the data is stored here. Okay, so as you can see, right, we have these two BW files. Okay, one is input, another is the ChIP-seq experiment. This is something that we have downloaded. So this is something that we can do, right, if we are interested in some sort of analysis or some sort of data exploration of already published data sets, we can download and process them. Okay, so the first thing we will do, as I said, is we will convert this, okay, from bigWig to bed graph. Okay, again, this is another tool that is available, and I have downloaded here. As you can see, this is the bigWig to bed graph format. Okay, and this is the command is okay. So bigWig to bed graph, this is the again a command line tool. So we are just calling this here. Then we have to give the file name, right, that we want to take and convert to bed graph format, and the output file name is here, which is input.bedgraph. Okay, that's what we are doing. Okay, so that's the first step. Okay, and we'll do that same thing for the actual ChIP-seq experiment here. Okay, so we'll take this K27me3.bw file and we convert to bed graph format. Okay, so it might take a bit of time again, so to generate this bed graph. Okay, so once this is done, then we'll go into R, right, and so in the meantime we can look into the R file, and we'll see again it looks pretty similar as before. We'll have to use this library PS genome musculus UCSC MM9, right, MM9 version that has been used here. So this is something that's very important because this method actually used this MM9 genome build. Okay, so we have to use the same genome build, otherwise we cannot really kind of compare what we are seeing here to their published results. Okay, so once we do that, right, we'll so this is done now. We have the bed graph formats. Let's see if we can see. Okay, so here you see this bed graph format, right? We have this chromosome 1, then we have these locations in the chromosome, and we have this counts or the scores, right? So that we have similar scores we got when we actually did the binning in our earlier data set. Okay, so this is again we have here, and similarly we have the same thing for the K27ME3 experiment. Okay, so we have now generated these bed graph files. Let's now move on to R. Okay, and let's start analyzing this data, and let's do the visualization. Okay, okay, so here we are. Now I have moved into the folder where we have this data set, and the first thing we'll do is we'll use this library as before, right? In the earlier case it was Drosophila library, right? Here it's mouse genome, right? We need the mouse genome. This is from bioconductor again, and we get the genome information. We store the genome information in this genome variable, and then the sequence length of the genome is given. So for chromosome 1, this is the length, chromosome 2, and so on. Okay, and we'll get this genome sequence. Okay, and we have the chromosome 1, 2, 3, etc. all these things here. Okay, so all this information, again the sequence lengths are given. Okay, so this information is, we already have the chromosome added, right? So we don't need to again add these two chromosome names. Okay, so we don't need to add this because this is already added here. Okay, so what we are going to do next is we'll again use the Biomart package, right? The library Biomart and the GViz package. Okay, this is something again you are familiar with. We did last time as well. In the last data set we have Biomart and GViz. So Biomart will connect to the ensemble using Biomart. So here you see ensemble mart ensemble data set, right, and the host name. Okay, so one of the things we have to be a bit careful about, right? So we have taken this HTFC May 2024, but perhaps what we have to do is here, as I said, is we have to look into these archives where we have this MM9 version. Okay, so this is something that's probably important here because we are looking at into the MM9 version. So this is actually in M38. So I think the version before this one, yeah, this NCBI M37, this is actually the MM9 version. So we should use this May 2009 archive, but maybe that will that might give us some error because of this archive is a bit old. Okay, so once this is done, right, we can also see whether we can load the 2009 because the annotations might be slightly different. Okay, so many of these annotations actually change depending on the which version that we are looking at. For example, some genes, their location might change depending on which version we are looking at. But anyway, it doesn't really matter because we are not looking at specific gene. We are just looking at a specific location in chromosome 2, right? So we are not really interested in any gene because gene location might change. Okay, so it says like there is an error. So maybe there is some sort of issue with the internet connection. I'll try again and see if we can load this here. last time we tried this twice and it worked. Okay, so once we have this, these commands will look very familiar to you, right? We have the feature map as before. We use the feature map symbol, which would be ensemble gene ID. Then we define an interesting location. So here we are not looking into any specific gene. We are just defining an interesting location and say, okay, we want to look at this position in the genome and see how the distribution of reads, etc. looks like. Okay, so that is something what we are interested in. So it doesn't really matter whether we are using May 2024 or 2009, but in ideal scenario, if you are interested in location of specific genes, their locations might change depending on which genome build you are using. So that is something you need to be careful about when you are loading this biomart ensemble in using biomart. Okay, so once this works, we can define this genome region track, and then we also can define this UTRs or promoter regions, and then we can load this data. Okay, so turned out there is some issue with the data loading. So what I will do is, let's first try with the data loading part, and then we can try again to load the biomart track. Okay. So we are loading the BW file here. The commands is import.bw, right? So for bed it's import.bed, and here it's import.bw. Okay, so let's load the data. And once we load the data, then before I go into this genome axis track, because we need these data track definitions, we'll define first this we'll load this biomart ensemble using biomart. So let's try this again and see if it works. So because we need that for visualization, and then we define, so for example chromosome 2 or chromosome 7 doesn't really matter. We define an interesting location. So here we are saying it's chromosome 2, right? So we'll define as chromosome 2 here for all of this. Okay, so once this works, right, we'll define these tracks and we can then visualize the results. Okay. Okay, so let's see. Again, let's see if the connection works. If we can load this archive, or maybe we can then also, if it doesn't work, we can try, for example, the other ones and see if they work. Okay, so this time it worked, right? So something to do with the internet connection or connecting to the database, and then we define the feature map, and then we can define this interesting location, right? Again, it depends on whether you are interested in a gene or looking at specific genomic region, right? So you can define as we like, right, whatever we are interested in, and then we are defining this gene region track. This is the interesting location, and then we can also define, we can look into this list attributes we have seen before as well. Okay, so we have seen this before as well, ensemble gene ID, etc. What are the attributes that we have? The chromosome is chromosome 2, right? And we can define the UTR region, which if we are interested in the promoters, right? So this is something very similar to what we have seen before, right? So this is we did for Drosophila in the earlier exercise, and this one is for mouse. So once this is done, right, we have now loaded this bigWig files here. We have the input and the ChIP-seq data. We'll then use this GViz library to actually define the tracks, genome axis tracks as before, and then we'll visualize the results. Okay. Okay, so let me see, right, this should work, and then we can go ahead with the defining the tracks and visualization. Okay. Okay, so this is done. So it's probably not getting the data anyway. So there could be some issues with the connection anyway. So we can go ahead with the visualization. We do not need to do the ETL definition anyway. Right. So we have now loaded this ensemble gene database for mouse, and then we are now starting with the genome axis track. Okay. And as before, we can try with this like a simple plot track function. So which will give us this location of these genes here. Okay. So as you can see, we have these locations of these different genes. Again, these are the ensemble IDs, gene IDs. You can look up which gene is what, right? Doesn't really matter to us because we are not focusing on any gene right now. The next step is to define these data tracks as before, right? So here we have the first data tag is for the input file. The second data tag would be the actual ChIP-seq experiment. Again, the commands are very similar. It's chromosome 2, right? We have control, and then we are putting the limits here. Again, we can play around with the limits and see what would be the actual limit, right? The actual appropriate limit. Okay, so it's actually loading, and then once the control track is defined, then we will go into the actual H3K27 track, right? This is the ChIP-seq experiment. Okay, we are defining that again in a similar manner. The genome version is MM9, and the colors, etc., which chromosome, etc., all these things are defined here. Okay, so we have done that. Now we can plot these tracks, right? So we can try this, for example, just the control track and see what happens, how it looks like. Okay, so this is the control track, right? As you can see, we have these gene locations, and the control track looks like this. Okay, so count data, and now we define this. We include the experiment track as well, ChIP-seq experiment, and see if we can find some enrichment compared to the control track. Okay, so as you can see here, we see some difference. For example, here we see a significant enrichment compared to the control. Right here, the score is actually higher than the control, much higher. Right, so we can see these small peaks as well, along with that really big one. Okay, so we can do similar kind of analysis. We can try to identify which regions are these peaks in, whether they are overlapping with promoters, etc. The other thing we can do is we can also do a peak call, right? So as you remember, these are not actual peaks; these are actually the counts, right, that have been obtained after binning, right? That is something we have to look how this where this like at which stage these bigWig files have been generated or written, right? But usually, as I showed you, that this is after the binning step, which we actually stored the data, right? So this is bigWig read density files, right? So as you can see in the data, actually this is bigWig read density file. So this is, we need to do a peak call, right, if you want to see the actual peaks. So it is possible, as I said, to do peak call from these bed graph files that we have got using this max3. There is a function called PDG peak call. Okay, so this function can actually do peak calling, right? And this actually can take an bed graph file, and it can actually generate or can identify peaks. Okay, so this is something that is possible, and we can very briefly explore. Okay, so we have visualized the data, so that part is done. We can see if we can manage to do some peak calling, right? So I'll do this. I'll show you, right, the commands. Okay. Yeah. So, this is the command max3 bdg peak call minus h, and this will be again in the terminal. Okay. So, in the Linux command line, so instead of peak call we just will say bdg peak call, right, from bed graph. Again we have these options minus I file, minus C cutoff, etc., and min den, max cap, etc., right. What should be the default values, or whether you can change, right, what is the minimum length of peak, right, or what should be the maximum gap between significant points in a peak, right. Again the default values are given, right. So, we can try this out with different parameter values and see what kind of results we get. So, as I said, right, if you use this program to do peak calling, you have to optimize the parameters. Actually, you need to do an extensive parameter optimization before you get some reliable results. Okay. So, this is something again not preferred, but just for your demonstration we can do this peak call for one of them, and we have this, let us say, minus I input bed graph, minus O input narrow peak, minus C, and minus L minus G. So, I have defined minus C is the cutoff, right. So, there is a default cutoff value we can, or default cutoff value we can use, for example, then another type of cutoff, right. Maybe you can say we can keep it default, for example, right, just keep it 5. So, which means 10 to the minus 5 will be the cutoff value for peak. Okay. We can try different ones. So, let us see if we just do a run and see what we get here. Okay. So, it is a similar process again, right. We run through these steps and we will do the peak call, and once we have this, right, is we can actually read these narrow peak files using this command in R. Okay. And this is the command, right. So, for example, they are like the bed format, but there are some extra columns. Okay. So, when we read this, right, we need to first narrow peak file, we have to define what is a narrow peak file, right, what are the fields that we have, for example, signal value, extra values that we have. Okay. So, I can show you in a moment what these files are. So, okay. So, we have run for the input and we are running for the K27ME3 as well, and we can have this here. So, as I said, right, this narrow peak file. So, the output that we are getting are in narrow peak format. They are in same, first columns are same, and then we have first few columns, and then you have extra columns. These extra columns contain this signal value. So, there will p value, q value, etc. So, let us look at this here, and maybe okay. So, as you probably remember, right, we have the first few fields, right, that are exactly similar as the bed format, but then you have these extra columns here. Okay. So, we have this, for example, and these extra values are here, right. So, for example, signal value, which is numeric, you have p value numeric, q value numeric, etc. So, all these things are defined here in these extra columns. Okay. And you have, so we have 1, 2, 3, 4 extra columns, and these are the extra columns, last 4 ones. Okay. So, you have p value, q value, etc., and what are these extra columns? Okay, peak and etc. So, we can read these narrow peak files in R using this command such as input.narrowpeak, right, and the format equals to bed, but with extra columns, which extra columns are defined here. Okay. So, we can actually read this in R. So, I will just show you that we can read them in R, and once you have read them, you can actually now define the tracks, right. You can define these different read tracks, and you can add these read tracks in here. Okay. Very similar way. So, we can perhaps try. So, maybe C1 peak tracks is equal to data track control narrow peak, and the rest of the things will remain the same, right. Okay. So, rest of the things we have to actually define here as before. So, actually they will not be data tracks, they will be annotation tracks that we defined before. Okay. So, you can try this out with our program that we used in the other data set, right. We have to define this as annotation track, not data track, and we can put them here along with this graph, right. We can visualize them here along with this graph. Okay. So, we can create this again from once we have loaded this narrow peak data, we can actually put them here in this visualization in R, right. We define these annotation tracks and we can put them as box as before, right. I am not going into that file, we can actually put this back. So, what you will see is like some of these will not make much sense, because again we have not optimized the parameters when we are doing the peak calling. Okay. So, that is why we always prefer to start with the BAM files, right, starting from the raw data BAM files and then finally to the peak calling part. Okay. So, we have now looked into the two data sets here, and I will go back to the final summary, right. So, we have now looked into this bigWig or BW files. So, in this exercise we have converted these bed graph files to bigWig files or BW files. So, this is something that is useful when you want to deposit your own data, right, analysis that you have done and you want to put that data somewhere. And then we have seen how we can actually process the BW files that are publicly available. We can convert them to first to bed graph, and then we can visualize this read density, right. So, that is something we can do, but as I also showed you, right, we can do some peak calls, right, using the MaxC program, but these peak calls, they require a lot of parameter optimization. So, we usually do not prefer them. So, to summarize, this week we have looked into ChIP-seq data analysis, and we have worked on two data sets hands on, which have given you extensive overview of how to process ChIP-seq data. Thank you.